Cell membranes, like the ones around these two cheek cells, are very busy places. Membranes regulate traffic in and out of cells, such as nutrients and important molecules that need to get into the cell for it to function properly, as well as trying to get rid of wastes out of the cell so it doesn't end up being poisoned by toxins. Membranes regulate traffic in and out of cells through the phospholipid bilayer, and these phospholipid bilayers, which form the membrane, are what's called selectively permeable. Selective meaning they choose what comes in and out, and permeable meaning being able to pass through. Because of this selective permeability of membranes, you end up with something called a concentration gradient, which is a term that just describes the difference between two neighboring regions. In a concentration gradient, a solute is more concentrated in one region than in a neighboring region. So a good example of that is when you put a tea bag in tea. You can immediately see concentration gradient when you first place the tea bag in the cup of hot water. Near the tea bag, there are many more brown tea molecules than anywhere else in the cup. In this particular instance, the solvent is the water that you're dissolving your tea molecules into, and the solute is the tea molecules themselves. Over time, the brownish colour spreads throughout the water, your solute, to create a uniform brew. And this is occurring because of a concentration gradient where energy is being released from an area of high concentration to low concentration, which is referred to as dissipation. And this is a fundamental concept for how cells regulate substances coming in and out of them. So all forms of transport across membranes involve gradients, and there's several ways in which cells can actually move substances across gradients. The three we're going to specifically be looking at are passive transport, which requires the release of energy, active transport, which requires an energy cost. You actually have to use energy for that to occur, and endocytosis and exocytosis, which also require energy for them to occur. So the first one we're going to look at is called passive transport, and this requires no energy at all. And diffusion is an example of passive transport. All substances are in a constant random motion, and the molecules do not stop moving at equilibrium. They can move in and out of your membrane still. There's three specific categories of passive diffusion we're going to be looking at, and those are simple diffusion, osmosis, and facilitated diffusion. So simple diffusion across a membrane doesn't require any proteins that are actually embedded in your membrane. They can pass straight through your phospholipid bilayer, so they don't require any carrier protein molecules. This only occurs with molecules that can freely pass through a membrane, such as non-polar molecules, and oxygen and carbon dioxide are classic examples of that. And you can see in this image here, this is how you basically can get oxygen back into your bloodstream through your alveoli in your lungs, and how you can get carbon dioxide back into your lungs to exhale. That occurs all through diffusion, where you breathe in, you've got a higher concentration of oxygen in your alveoli than you do in your capillaries, which have got deoxygenated blood, and so the oxygen diffuses through into that capillary. Likewise, the carbon dioxide, which is in higher concentration in the capillary, moves back through to a lower concentration into your lungs, and you can breathe out the carbon dioxide, which would otherwise be toxic. And these are examples of substances moving down their concentration gradients. You could ask, how do cells maintain these concentration gradients? Otherwise, wouldn't your oxygen effectively move into that capillary, and you'd end up with an equal concentration on both sides, and you would never end up moving any more oxygen in? Well, what ends up happening, of course, is you actually consume that oxygen as you use it to respirate, and therefore your cells will maintain that balance of concentration gradient, which you require to be able to absorb more substances. Now, osmosis is a specific type of diffusion, and it refers specifically to water and moving water across a selectively permeable membrane. There's three specific terms you need to know about when we talk about osmosis. One term is isotonic, and that's when you have equal concentrations of water inside and outside of a cell. In that particular case, your water molecules are still moving back and forth, but the net movement, or the overall movement of molecules, is zero. Hypertonic solution is when the solute concentration is lower inside the cell, and a hypertonic solution is where the solute concentration is higher outside the cell. And this specifically relates to what's called turgor in plants. And turgor is when you have enough water in a plant for it to stand upright and not wilt. To illustrate this, you can do a prac where you use a U-tube which has a selectively permeable membrane between both sides of the U-tube. It's selective because it allows water to go through, but it doesn't allow a solute molecule to go through the membrane. So that solute molecule could be sugar or salt or any other thing that dissolves in the water. So in this particular illustration, you can see that on one side of the U-tube, you have a low concentration of solutes. That side is hypotonic, whereas on the other side of the U-tube, you have a higher concentration of solute molecule, which makes that side hypertonic. In that instance, the water wants to move from the hypotonic side to the hypertonic side, resulting in the water on the right side of the U-tube increasing. The same principle can apply to blood cells. You can see these three images here. On the one on the left, you end up with an isotonic solution in which the solute is equal on the outside and the inside of the cell, and therefore water is moving in and out of that cell at the same rate, giving you a net equilibrium. In the second illustration, you can actually see that it's a blood cell in hypotonic solution, and in this particular case you end up with water going into the cell, and that can actually cause cells to burst, or lyse is another word for that. In the final image, the blood cell is actually in a hypertonic solution, and in this instance you actually get water moving out of the cell and effectively dehydrating that cell, so you can see it's losing its shape. And these arrows indicate that there's actually more movement of water out of the cell than into the cell. The same thing can apply to household plants, and this is something you would have seen on a regular basis. In a hypotonic surroundings, the cells of this plant have plenty of water moving into them and giving the plant good turgor. In other words, it's able to stand upright. In hypertonic surroundings, the opposite occurs, and you end up with wilting occurring. Facilitated diffusion is the last type of diffusion I'm going to refer to in this video, and it requires proteins that are embedded in your membrane. The membrane proteins actually assist the movement of a polar solute along its concentration gradient, and that's really important because a lot of those polar solutes can't pass directly through the phospholipids themselves, and glucose is a classic example of that. It's too hydrophilic to pass freely into red blood cells, but it's absolutely necessary for us to survive and to be able to get energy to perform any of our actions. So that's a classic example of that. In summary, what we looked at was passive transport, these two illustrations on the left here, which require no energy. We looked at simple diffusion, where you end up with molecules moving from an area of high concentration across a membrane to area of low concentration. And we also looked at facilitated diffusion, which does the same thing but requires a transport protein. Something we're going to look at in the next video specifically is active transport, and this requires energy.